seems it doesn't show the board. And this, yeah, it does. So last time we've ended at discussing post-translational modifications, and we have to sum up all the post-translational modifications that can happen at certain amino acids. And there is a small table from Nature published where all different amino acids, they are here by only one letter code. And here you have all the modifications that can be found in human proteome. And as you can see, there are some modifications which are quite frequent and some modifications which are rare to non-existent. Why we need this table and what this table can help us to do is if we are trying to work with some protein, and we know that this protein contains some, let's say, some of these amino acids, we can get some, we can have some guess about us what is the possibility of post-translation modification, and also if we are working with some proteome of, for instance, human or mice, there is already some annotated form where you can just find which amino acids are modified more frequently in this given protein. So you can just open the database, enter your protein name, and there will be not only the sequence of this protein, but also some more frequent protein forms. So if you are working with some of these proteins, you can already know what proteoforms you can have in your sample. But if not, you can use this table to see which of proteoforms can be here. For instance, if we have serine here, as you can see, there's a really high chance of having relation, for instance. And this table can help you to do all you need with proteoforms and also with identification of proteins. Because just in the next lecture, we will start with mass spectrometry. Mass spectrometry will allow you to see the mass either of the protein itself, which is not quite informative, but also mass of small peptides from this protein. And you will be able to use different proteases to cut your big protein at some certain locations, and then you have to search to the database on the molecular weight of your peptides and molecular weights of peptides which are in this database, either in database which was already pre-processed or in a FASTA file, because if you have a FASTA file you can convert it back into proteins, you can cut it in silica, and also have some ideas of peptides which can arise from your protein. So, if there are some of those modifications, then the weight of your peptide will be different. And for some modification, it's obvious, like phosphorylation or so, but what if you have 2 or 3 different modifications? It would be a trouble, and you would like to know whether the modifications are possible and try to search for peptides which are mostly or not modified, or vice versa, if you are more interested in modifications per se. As we've discussed before, those modifications can have a detrimental effect on health, for instance, right? So you can try to choose such peptide from your protein which carries this modification and analyze only this peptide alone. And if this peptide is unique for the whole protein, if it's only for this protein and none of other proteins produces such peptide, you have everything to work with this peptide, right? So this table is actually quite useful. I will skip this one, and we also have to discuss that we have interest not only in endpoint concentration of different proteins per se, but also we are quite interested in so-called turnover, because what we have here, we have synthesis of protein and degradation of protein. And we can get more protein by either decreased degradation or increased synthesis. But as you probably know, that most of those processes of synthesis of a new protein is actually controlled by its availability in that cell or availability of its substrate, or somehow it is controlled. And what can happen in your cells? That this endpoint concentration will still be the same, but it is probable that both synthesis and degradation will increase. What do you think? White cell could require, economically it's not feasible, right? To make more protein and degrade this protein also faster. What can be, what benefits cell can get from such strange thing, for instance?
Maybe the cell needs this protein for a short time.
Right, for regulation. Because if you have only small amount of water entering this bus tube, and small amount of water which is draining from this bus tube, if you want to fill it or drain completely, you will have to wait a long, long time. And if you have a lot of water streaming down to this bus tube and big hole in this bus tube which drains this water, you can get regulation really fast. So for some proteins and some processes, turnover is more important than an endpoint concentration of a protein. And we actually can study this protein turnover by labeling some proteins which with some chemicals or with some stable isotopes. You know that, for instance, with nitrogen, we have two stable isotopes. One is the most frequent one, more than 99%, and the other one, which is 1 dalton more in weight, is rarely occurs in nature, but we can actually use protein synthesis for labeling our protein. For instance, if we have cells from, let's say, vertebrate animals, how can we make this cell to incorporate this nitrogen, heavy nitrogen, inside newly synthesized proteins? Yeah, my idea was that we must be interested in turnover of protein. So we need to ascertain how much old protein was degraded after some time, how much of new protein was synthesized in a cell. And we can label newly synthesized protein by incorporation of heavy nitrogen. Nitrogen isotope which is 1 dalton more. So, taking into consideration last lecture on protein synthesis, how can actually we make our, let's say, vertebrate animal cell to incorporate this heavy nitrogen?
To capture them in the medium with only heavy nitrogen.
But will those cells use this nitrogen only for proteins, and will they synthesize all the proteins they need, and will we actually get the final result to this? Or there is some other way, some proteins which are not synthesized by vertebrate animal cells. There are some proteins which are just some. Some proteins are not synthesized by at least mammal cells, but some of them even invertebrate cells. Only prokaryotes and plants can synthesize them. So we can add those amino acids inside your medium, not nitrogen itself, right? Because it's not quite interesting enough. But if you have those essential amino acids which are not synthesized by this organism, any act of incorporation of this amino acid will also label our newly synthesized protein. So this way we can actually get some information on a turnover of a protein, and since all the proteins actually have to go through this cycle, synthesis, integrating labeling, then proteasome degradation and so on, we have two different proteins. Well, the protein is the same, but the mass of this protein will be a little bit different. One of newly synthesized and other is whole proteins. And actually, we can then, sorry, I have to get here, using this isotope of nitrogen, then get those proteins separately. For instance, if we'll do some mass spectrometry and also we have some peptide which is unique for some protein of your interest, you will have those peptides differ by this mass of 1 Dalton here because of this nitrogen 15, and you can have both of them on the same run of your mass spectrometer, and you can just compare the height of peaks or actually the concentration of old and new protein to make some deduction. And what we have here is that there are different scenarios, either of the protein which doesn't change its concentration during our experiment, but probably turnover is different, or protein can change the concentration, but due to different processes, either more synthesis or more degradation. And actually, it's a different behavior for the cell. And if we are searching for some, let's say, biomarker, for instance, or even some target for medicine, we have to know this information, what had happened due to our experimental conditions, right? So what can happen is here, but what I want to point here is this graph. As you can see, there is cells and there is a logarithmic scale of newly synthesized protein level and of total protein abundance. So this axis is how much protein of certain gene products are here in cell, and this axis is was this protein newly synthesized from an old one. And you can see that most of the proteins have the same level of synthesis and degradation. It's nearly at the center, right? And only some of proteins change its turnover, and some of the proteins doesn't actually change its endpoint concentration, but turnover actually was changed. And we can plot such information and actually then see what were those proteins which change their turnover rate for different purposes. So, why it is important for cell to make such a stupid thing? We already proposed that this is a way of control the concentration and be able to change this concentration faster. And what we have here is such a graph, almost as the previous one. We have logarithmic axis of transcription and translation of the same gene product. And as you can see, we can get the same amount of protein by either have a lot of translation and just some mRNAs here, so just some small number of transcription, or vice versa. We can get a lot of transcripts, but only some rare events of translation. And this line is equi-concentration line. We have the same protein synthesis, but as we will see later, it does have an impact on cell, and it also does have an impact of our synthetic constructions that we usually use with the cell. For instance, we routinely in our laboratory produces a transgenic organism which synthesizes some protein. For instance, green fluorescent protein for labeling some protein product. It's actually a transgenic bacteria or animal or whatever. And the problem is that if we'll try to see this graph for different organisms, for yeasts, for mice, for human, for E. coli, as you can see, most of the proteins have some maxima here, where there is some transcription, some translation, and it's somewhere in between, right? There's only rare cases of extreme cases where there's a lot of transcription but only a small amount of translation, or vice versa. And what we have is mostly our synthetic constructs, as you can see, this. So we have to think not only about the efficiency of our promoter, if we are constructing some organism, because, well, we have some promoter-formed cytomegalovirus, for instance, and we have a lot of mRNA, but there is no guarantee that this overwhelming quantity of mRNA ever get to protein, right? And if we also have some untranslated region engineering and so on, we can get to this area where actually most wild-type proteins are present. If something is not quite understandable, just ask, because I do understand that this is rather not quite an easy topic, but was this understandable at least? All right. Then, as you can see, there is actually some of the proteins which are here, which have, for instance, a lot of transcription, but only some amount of translation. What do you think about these proteins? What will be characteristics of such proteins which have such an unusual ratio between translation and translation? Transcription and translation, sorry. If there is quite a small amount of protein, right, will it be possible for that cell to actually control this quantity of this protein precisely?
It's, if there are a very small amount of protein, a cell can understand that it should express them more than now.
It can be the case of some dysregulation, right? But actually, those proteins are synthesized at some background noise level. They are quite rare, right? The cell can control them. And there are some proteins which actually are like sensors or something which need to be in this region. But most of the proteins are here. And let's see that we have this line graph, which says that those proteins are more or less stable and we can control them, varying by the speed of control, of course. But this part of this graph is something that is more or less at a noise level for the cell. And also, what is interesting here is that we can plot such a line of equiabundance, the same level of protein, but cell can either have by a lot of synthesis and a lot of degradation and a lot of control on the concentration of this protein, or cell can be more economical, provides only some mRNA and have a lot of translation from this mRNA, and of course it will be possible to control it in an efficient way. But for instance, for housekeeping genes, do we need such tight control? If there would be 5% more, 5% less of this abundant protein, will it have some bad impact on the cell?
Housekeeping.
Yeah.
As we said in previous lecture, there are a different level of housekeeping genes in different tissues and even different housekeeping genes.
Yes.
So it can hurt.
Usually they are overabundant.
Yes.
Right? So 5% less, not a big problem. But for some proteins, we have to have them really in tight control. So we can plot such lines where we can have either coefficient of variation, so our control on this quantity of this protein, or fitness of cost, whether it's economically feasible to have such ratio between translation and translation or not. So only some proteins which need really granular control on its concentration will be here. All the other proteins will be somewhere there. So, what we have to conclude here. If we are engineering some new cells, some transgenic organism to have some protein, actually what we mostly do is we concerned with transcription, right? So here's our line. Here's our line which says that it could be abundant line. And what we are trying to do most of the time, we just write transcription and we end up in this region where the concentration of this protein could vary a lot. And if it will be possible somehow to control both transcription and translation by cleverly engineering our construct, we'll be able to stay in this part of our graph. So now we know that turnover is also important and we need to study it. And let's see what can happen if this ratio of turnover changes. For instance, we have cell and we have some stress, let's say heat the cell or increase the salinity or whatever. And it's all the possible ways how turnover can change inside the cell. So the green one is newly synthesized protein, the red one is pre-existing protein, and the blue line is a total protein. Here we have some time, because it couldn't be instantly changed by the cell. The cell have to synthesize this protein, and it will take some time. And here we have, as always in physics, where there is some process which tries to get to some value but never reaches it. We use half-life of this protein, for instance, and here half-life of reaching this point. And what can happen, as you can see here, degradation can be higher or lower, or stays the same, and synthesis can be lower or higher, or stays the same. And getting just this point will answer the question what will happen later with our cell and what is going to happen, right? So, how can we do this? We can label our cells, as we already discussed, with some heavy isotope labeled amino acid, and then have analysis of your proteome as a whole, or some protein in this proteome which you are interested in, and sample it at some time points. Of course, at some zero time, even if we already change the medium, there is no heavily labeled, heavy isotope labeled proteins, 0 here, and 100% of proteins are without label. Then at some time we get different results, and of course, if we have this point, we can calculate what will happen. As you can see, for most of the proteins, half-life is about 5 to 6 days for human neuron cells. And of course, there are some proteins which are here to stay for longer. For instance, cytoskeleton proteins and some other proteins, histones. Of course, they are not quite often disassembled and synthesized in you. They sit on DNA and they won't be easily degraded if cell is in a stable condition. So those numbers which I was giving you at the start of lecture about the half-life of protein, as you can see, there is an experimental data which confirms it. Also, let's see which proteins actually increase turnover and decrease turnover if we change, for instance, some environmental factors. As you probably know, for some neurons, there are different receptors which can sense different mediators. And also there are some receptors which are so-called ionotropic receptors, which can regulate permeability for some ions. And here we have just a voltage-dependent channel, sorry, let's say right here. Sodium channels, right? Yeah. You know, for sodium channels, there are specific substances that can block those channels, like tetrodotoxin, for instance. Do you remember this from the school program? So if you use such a chemical on a cultured neurons, you can see that there are some changes. Some proteins stays almost the same, but some have a lot of change, either having more protein, like coupled protein, and some proteins which are needed for synthesis of new mediators, and also some proteins which are dysregulated in this manner. They have lower turnover, and actually some subunits of those channels are at a higher turnover than in a steady state. So as you can see, such studies of turnover can provide us with some information as what happened to the cell if those channels, for instance, are broken, blocked, not working, or whatever happens to them. So let's continue then. And the last application of proteomics is so-called reverse genomics. Because let's say you have some organism which is not yet have annotated genome. And for the sake of having even more trouble, let's say this organism is a plant organism. Why plants have a lot of so-called pseudogenes. And plants just like to copy the same gene many times. And you don't know what copy is working, whatever. And if you will try to compare genomes of plants at nucleotide level and at protein level, what do you think at which level those omes, protons and genomes, will be more similar? Whether protein products will be more similar or just a sequence of amino acids in DNA, and why?
Be similar.
Let's say we have wheat and rye, different plants which we use for food, right? And 2 years ago, rye wasn't actually sequenced yet, and even now, rye genome is not quite annotated. And wheat genome, as you can understand, was one of the first to be sequenced, quite, probably not fine, but quite good. Annotation exists, and so on and so on. And we want to annotate the genome of rye using some material from wheat. Of course, you could do this at genomic level, right? But there is a lot of of those pseudogenes, and you don't know which of them are actually what. Of course, you can get to transcriptome level and get transcripts, but also not all transcripts lead to some protein products. And what would be better? To compare some gene of unknown, let's say, function in rye at nucleotide level to the same gene in wheat, or at protein level?
Both.
We have synonymous mutation, which is also called silent mutation, which codes for the same amino acids, right? And since there will be some mutation, and if those mutations are not do not have quite significant influence on, let's say, life of this plant, they will persist, right? But for proteins, if you change some amino acids or some conformation, mostly this protein will be either worse as a function that it is doing, or it will be dysfunctioning at all, right? So the protein structure will be more conserved than genomic sequence for this protein. Do you agree?
Sort, yes.
So if you want to search similarities, it's better to translate it to protein, compare proteins, even in silico. Actually, most programming for automatic annotations do like just this. They try to find open read frames, translate all possible products, and compare them, and so on, because they will be more similar, and then you can get their growth. And also, even if there is some organism which is not sequenced yet, but you have some close relative of this organism, you can actually sequence this gene by this genomic. You have the sequence of your amino acids here. You have a triplet here. Of course, this code is not one-to-one equal. There is more triplets than there is amino acids, but you can actually use so-called mm, again. You can synthesize primers which have different nucleotides in some positions. So they would be able to act as primers for sequence with one or two different nucleotides. And those primers, sorry, just forgot the term in Russian, degenerative, right, absolutely, sorry. So those degenerative primers can be synthesized by knowing just primary sequence of your proteins. And since we already know that at protein level, most of our proteins with similar organisms are also similar, we can hope that our primers we have just designed can allow us to get this part of genome, even the whole gene of this different organism. And we do not have to wait for genome sequencing or genome annotation or even transcriptome here. It's possible to do by, so reverse genomics is also one of the applications of proteomics. So that concludes our first lecture, and what we are discussing actually so-called Proteomics 3.0, which includes not only the protoforms or proteins, but also abundance, turnover, PT, post-translation modification analysis, isoforms, protein-protein complex integration, localization, and so on. So nowadays proteomics is from microscopy, optical and electron microscopy to NMR analysis. Every method which is available we can use. So let's continue with our first lecture on actual methods of proteomics. As I've already told you that proteomics itself doesn't have a lot of methods. Mostly we use methods from bioinformatics, protein chemistry and so on. And today we'll be discussing some methods from protein chemistry. But actually, if we are doing just our electrophoresis, one-digit electrophoresis, we don't see just one protein. We see all the proton. So we can actually say that this gel-based methods also proteomics methods, right? And what we want from this method, if it is possible, to get as good a separation as possible, to see every protein, every proteoform as a separate point, band, or dot, right? That's actually our idea. And what is also nice about gel-based proteomics, we can stain our gel and we can calibrate and get result of the concentration of our protein right from the gel. We don't need some special labels, some special mass spectrometers which can analyze without this label and compare, because mass spectrometers are actually not quite quantitative. They are qualitative, right? But quantitative is not so good. We will be covering it just next week. So, but before we can make our gels, we have somehow to get all the protein, to extract all the proteins from our samples. And we have a lot of problems here. First, some of the proteins are hydrophobic, some hydrophilic. Some proteins form complexes. Some of proteins are have complexes with nucleic acids, with lipids, with whatever there is. And with DNA and RNA, it wasn't such a big trouble, right? They have some properties which allow us to get whole DNA from cell, or RNA, of course not quite whole, but deal. But with proteins, it's another problem. And what we need to do in order to extract this protein, we have to somehow solubilize all the proteins to make them soluble in mostly water or some water-based buffer. Also, with DNA, it's not the case, but with RNA, we have RNases, which starts to degrade your RNA right away. With proteins, we have a lot of specialized proteins which modify different proteins as well. First of all, we have proteases which cut the proteins. We have some phosphatases which get some, which work with. We have kinases. Kinases, phosphatase, of course, can get rid of or add some additional phosphorus, and so on, so on, so on. So for us, it's better to somehow to stop all those reactions from happening. And since we are working within water solution, and unfortunately most of proteases are hard to kill, we have to use some chemicals which can block those proteases and phosphatases mostly. For other proteins, it's not as easy to stop them from functioning. Only some way of denaturation can help us with it. So for us, it would be better to stop the processes we can and then denaturate proteins as fast as we can to be able to stop all the processes, all the enzymes from working. Also, if we have some contamination of DNA and RNA, it can hamper our analysis. First of all, some proteins form complex with nucleic acids, and we wouldn't be able to separate them because they would be on this, let's say, DNA or RNA, right? Also, if we have gel electrophoresis, then those long molecules, which are DNA and RNA, they have charge and they are long, and they can just block our gel from being able for proteins to enter the gel. We'll have a lot of how to say, we'll have a lot of proteins sitting at the bottom of our gel without being able to enter anywhere. So for us, it would be nicer to actually clean our sample from RNA and DNA. And also, we were talking about some animals, some cells, and almost every plant here. There are some metabolites which also will be a problem. For instance, in plants there are such metabolites as polyphenols. Polyphenols, by their nature, likes to stick to everything. For instance, if you are eating some non-ripe fruit which is green, yeah, you have this strange feeling of your mouth to become dry. Or if you drink a lot of green or brown tea, right? You drink water, but you feel like. And this is due to polyphenols. They like to stick to proteins, and what they do, they also form complex of proteins, but not a natural one, but just sticking them together. So with plants, it's total nightmare. You have to somehow get rid of those metabolites. For most of human mice, E. coli cells, it's not a problem, actually, but plants are a different story. Also, what problems we have? Chemical properties of protein, they are quite different. Protein solubility, not all proteins are soluble, and we have to make them different matrices. We already discussed it. There are some kind of additional secondary metabolites, for instance, and also different cells have different cell walls. For instance, if you are working with yeasts, they have this cell wall, polysaccharides, which you have somehow to break. It's either can be done with some harsh conditions like liquid nitrogen and you just grind it, or with some glass beads, small glass beads in a machine which just vibrate and break those cells and so on. For other cells, it's not a problem. For instance, for mammalian cells, you can just add some detergent like SDS or Triton X100 or whatever, and they will be readily accessible. And what we'll get at the end is a mixture, complex mixture of hundreds and thousands of proteins. Let's see what we will use in such a protocol of extracting proteins. First of all, let's talk about some uh, some protease inhibitors. There is a lot of different proteases, and we can't have just one inhibitor which inhibits them all. There is no such thing because of different nature. But we can use so-called cocktail of proteins, and I've get this table from Thermoscientific website. It's almost complete, which provides you with a list of mostly used chemicals, and some even not only chemicals. Protein is actually a peptide, um, which can block some of these proteins. What I want to stress here, some of them are irreversible, so they just bind to this protease and stay there. Some of them are unfortunately reversible, and if you will continue to work with your proteins and somehow dilute your sample, hence say you will dilute this inhibitor, then proteases can start working right in your solution unless you denature your proteins. So pay attention with those reversible inhibitors. You have to have the same concentration of this inhibitor um, unless you denaturate your proteins, right? Also, some of them have very small molecular weight and you can easily get rid of them, but some of them actually peptides of considerable molecular weight, and it can happen to stay in your protein mixture sample. So pay attention that some of them, especially if you are working with some small proteins or peptides, they can get you additional peaks on the chromatograms or on gels. Don't be afraid, it is possible to do so. And also there are some of them are like PMSF, which is really good serine protease inhibitor. Unfortunately, it's reversible. It is highly toxic. So some of them are really nasty substances. Also, some of them are soluble in water, and for some of them you will have to add at least some amount of methanol or ethanol to solubilize them. Usually some of them are in DMSO vial and you just dilute it in water, but some of them needs addition of ethanol. Of course you don't need all of them at once, because some of them have almost the same way of action. For instance, ABSF and PMSF blocks the same proteases, and you can use either of them. But there is, as always, some catch. ABSF is better, it's not toxic, it's, as you can see, water soluble, it doesn't degrade like PMSF, which degrade in water in, let's say, some hours. But if you will use this ABSF, there is a problem because it's an irreversible, it binds to proteins, and actually if you have this molecule, and at some charge, as you can see, there is this group and this group, and even if one group is used to attach to the protein, other group have some charge on it. And if you will do isoelectric focusing of your proteins, you will get additional proteoforms which are not present in your sample. As you can see here, upper protein here, and there is some 7 isoform of it, and you can see after using this inhibitor, you get 8 isoform. Right, so pay attention to the cocktail inhibitor cocktail contents. Usually you get some paper stating what's inside this cocktail. And if you will buy them, there is a lot of different cocktails on EpCam or Sigma Aldrich or whatever site you will be using. It's actually, you can check what is inside this cocktail and choose the appropriate one. For instance, if you are working with bacterial material, you do not need some of those inhibitors because there are no proteases of such classes in prokaryotic cells, and you can just use those three. You could either use ABSF or DMSF. But if you are working with mammalian cells, there are two additional classes of proteases, and you will have to add at least those two. So, also, if you're working with proteins which are metalloproteases or proteins which use some metal ions for functioning, you should not use ADTA in the solution because ADTA will actually chelate most of divalent ions, which is a good thing. And metalloproteases wouldn't be able to work because they have this metal in its active center. But it will also inhibit any proteins which depend on metal ions. For instance, iron, almost everything, almost every metal will be hampered. So, if you plan to use this for measuring activity or natural folding of your metalloproteins, then EDTA is a thing that you should avoid. So, we can skip this, I think. Also, if you working with proteoforms, the most common post-translation modification is phosphorylation. And there is kinases and phosphatases which can alter this post-translation modification. And we can use phosphatase inhibitors. Of course, they also have a cocktail of different inhibitors because there are different families of those phosphatases. There is acid phosphatases, there is alkaline phosphatases, and also if we're talking about, let's say, alkaline phosphatase, there are different forms of this phosphatase. For instance, in placental tissue, there are another group of those phosphatases which are not blocked by most of those inhibitors aside from imidazole. So, if you plan to work with post-translation modifications, you have additional plan to somehow quench those phosphatases. And what will happen if you will not use them? Here we have Western blot, which have some antibodies to a proteoform of special proteins, which is phosphorylated. So antibody recognize only phosphorylated form of this protein. And you can see that if you do it quite quickly, like 0 hours after separation, then you get well, not such a bad results. But even after 3 hours of just standing on the lab bench at 4 degrees Celsius, you already have some less amount of this protein because this phosphate is already gone. You have this protein, it is here, but phosphate is not here, and antibody was specially designed to recognize this phosphorylation event. So if you are working with post-translation modification, be extra careful to block at least phosphatases. But of course. Okay, we've successfully extracted our protein, and now we have to somehow analyze it. What we want to know, first of all, is molecular weights of our proteins. And if we'll just put our proteins in solution on gel and start doing electrophoresis, we actually will be separating them not by molecular weights, but by either electric point or electric charge, right? And it's not a lot of use for us. This information can be useful, but not so. With nucleic acids, this is not a problem because we have additional charge for every base, right? But for protein, we have to somehow add something that will homogeneously add charge to these proteins. Since proteins have both hydrophilic and hydrophobic amino acids, we can use so-called sodium aluminum sulfate, here as SDS. As you can see, it's a kind of lipid with this non-polar group, which have also a charge. And since it can easily bind non-covalently to our proteins and cover it more or less homogeneously, then we can say that if we'll have this SDS in excess in solution, then our proteins will be covered, and the longer the proteins, the more will be the charge here. So we have some relation between charge and the molecular weight or length actually of this protein, and it will start to do separation by electrophoresis after such approach, after using this SDS. We actually can get some ratio between this charge and the length of protein and get some ideas of the molecular weight of protein. Also, what SDS is about is that if you have a protein which is somehow folded and you add this SDS molecule, it will compete for those hydrophobic parts of the protein and protein will start to unfold. But what do you think? Will this protein unfold by itself fully at room temperature? Can it be possible that there are some parts of this protein which have which have stronger hydrophilic interaction between proteins and between protein and this additional SDS molecule, right? But what we can actually do is just heat our sample. If we heat it, then as you can know, the free energy would be the difference of free energy. It would be not enough to hold our protein in this folded position. It will unfold. And if we'll then cool our sample, it will be already covered by those SDS molecules, and it will hold, at least, will stay in this linear form, right? So to work with our proteins, we will have to use so-called lysis buffer, which contains some SDS, some buffer trees, some EDTA to chelate divalent ions, and we have to heat our sample with this lysis buffer to have our proteins covered with this SDS. But as we've told before, we have to get our SDS in excess of concentration of our protein to make sure that they are covered homogeneously, right? Of course, we can't just measure proteins for all of our cells, tissues, and so on. It will be different, but there is such a ballpark number which we can get by have an idea that about 10% of total weight of our sample is protein. And then, of course, for different samples it could be not the case, but for most of the samples it is the case. We can calculate probable protein concentration by using this formula. We have total mass in milligram, and this 10% is actually our weight of proteins and the volume of lysis buffer. And so we can get some protein concentration in our lysis buffer, and it should be below 10 milligram per milliliter. So you can actually calculate the volume of buffer you need to solubilize and to actually work with your sample. Just use this simple formula. So now we have our sample, and actually what I would like to suggest that if your sample is, let's say, plant sample which have a lot of proteases, for some sample which is hard to crush, use cryo wells. It's you don't need to buy some expensive ones which have this iron ball here, which just under nitrogen crush your sample. You can use just mortar and pestle with nitrogen, not a problem actually, and get results which is quite fine without using any technical methods. So actually this methods of just putting this solution, mixing it with our samples, works mostly for cell cultures, for some tissues, but for some more troublesome preparations, this method is not quite good. For instance, if you are working with plant cells, it can work, but most of the time it wouldn't get you enough protein, and also it wouldn't be clean enough. And you can use different approaches. Of course, all the concentrations and weights and so on are given here from a textbook, and if you are already working with some laboratory, you will have a little bit different recipes, because most of those concentrations are, well, they need to be changed for different samples, right? So don't take those numbers as a rule. Those numbers are just some approximate numbers which work for most of the samples. So we can use different lattice buffers. As you can see, we can have different concentration of SDS. We can even use different chemicals, like here we have vanadium hydrochloride. And one method which I want to discuss is the one that used in laboratory a lot. And this method can allow you to isolate both DNA, RNA, and protein. And you can get everything from your sample. But as with all pseudo-universal methods, it works for anything but mustard now. It's a trial method, which I'm sure you're familiar with when you are working with RNA extraction or DNA extraction. Have you already done this trial extraction method for nucleic acids? Okay, so triazole is actually some phenol and addition to this final phenol, and it you actually buy it like a solution in a bottle. Then you add it to your sample, homogenize it in this solution. This solution is good because it stops most of proteins from working. Most of enzymes will be in an active state, so sometimes even no inhibitors are needed. Then you add here some chloroform, stir it, and then centrifuge. This chloroform will have mostly RNA fracture of your biomolecules. So you already have RNA, and you can then participate it and work with it without any problem. Then you add some ethanol and do the same. And this ethanol will be your DNA, mostly. And what you have here, you can actually add isopropanol, and most of your proteins will just become pellet. You have to discuss supernatant, it's not a problem here. You have to wash your protein from phenol and other chemicals by using vanadium hydrochloride and ethanol. And then those proteins are insoluble because there is no water. And you have now solubilized them. You have to use cyurea and urea. Those agents work as chaotropic agents. They break hydrogen bonds, and it will allow you to solubilize your proteins. They will be in solution. And after collecting supernatant, everything that is soluble is actually most of your proteins. So as you can see, this method is quite nice because it allows you to get all the biomolecules we need for analysis. But of course, it works with everything but mustard now. So let's see some more troublesome protocols for plant samples, for instance. As we've already discussed, that plants have additional secondary metabolites, which can be a problem. When they are in a plant cell, they localized in they are compartmentalized and they do not mix with our protein. But when we crush our samples, when we open our membranes, everything gets mixed and those secondary metabolites as well. And there are some protocols which include additional steps like participating your proteins out of solution with trial chloroacetic acid and called acetone, for instance. So we already participate all our proteins, and only then, since they are participated, they couldn't work, and since there is not so much water, those secondary metabolites wouldn't also be able to work. Then we participate them again with methanol and ammonium acetate, and we get those to participate, and then we have to wash them. And to wash them, we use different lysis buffer, but also we add sucrose in it, just to make it possible to separate different phases. So phenol and this water with sucrose, or will be phenol at the bottom and sucrose at the top, we will be able to actually aspirate it easily. Because vice versa, you will have to punch this phenol and get your phase from down the north, which is not quite. Also, what I want to stress here is that the pH of phenol is of importance. If you already tried to isolate RNA, you know that pH of phenol can either allow you to get RNA in a phenol or to get rid of RNA in a phenol, right? Have you done any isolation of nucleic acids yet? So if you are doing DNA and RNA, you will get a different phenol. One is basic, other is acidic. It is the same phenol, and since phenol and water are not quite mixable, it's okay, but some water can actually dissolve in phenol, and you can add some buffer in this water and make this phenol of different pH. Right? So for this isolation, you need phenol at pH more than 7.8, and you can add bromophenol blue as an indicator that your pH is right. This phenol will be not brown, but actually blue at this pH. So, and then we'll have liquid-liquid separation, and all the lipids, pigments, polyphenols, proteins will be separated in different particles. For our proteins, they will be mostly in this solution, and we can again participate it, and just by adding some methanol and ammonium acetate, we have now participate of our proteins. We can wash it with cold acetone from lipids and from everything that is polar enough, and solubilize it. As you can see, the protocol is really close to one which we already discussed, the Trizol protocol and so on. So the steps are not the same. It's what you are using to get most of your proteins that is different. One question though, what do you think? This protocol and previous protocol, will it get all the proteins from your cell, or some kind of proteins will stay in solution with phenol, even in those conditions? As you can see, we have used methanol and ammonium acetate to precipitate our proteins. But what if our proteins are highly hydrophobic? Will they be participated from water phase by addition of methanol and ammonium acetate, or will they prefer to stay in the phenol phase? Right. So there is no universal method which allows you to get the whole proteome. And if you will be trying to get all the proteins, you will have to do multiple rounds of extraction, with different protocols, compare them, and probably we will have to do different separations for different proteins and then just combine those results. And if you want to get proteins from membrane, you will have to use a little bit different protocol, which will include mostly water-soluble part here, and you can isolate membrane proteins by it. So it's not necessary to memorize those protocols, but the idea is there is no universal protocol. And even if you want to get all proteins from your sample, one sample for instance, you probably will need two different proteins to get membrane-bound proteins and cytoplasmic proteins, for instance. And even if you are interested in some proteins which are highly basic, for instance, or highly acidic, for instance, you are working with histone proteins, we will also need to do additional round of different separation. So now we have somehow isolated our protein. Now we need to do some quality check here. What we want to know? We want to know whether our separation is clean enough. Are there only proteins or are there any other nucleic acids and whatever? Also, we want to know the concentration of our protein, how much of protein is there, because we need this concentration for downstream processing. And we need to know whether our proteins stay in more or less intact condition, or we have some protease or something that can just make our sample not available for downstream processing. So let's start with the first problem, is getting concentration. There is a lot of chromogenic assays which allow you to ascertain the concentration of protein by getting some colored product of reaction, such as Bradford assay, Pierce BSA assay, and so on. The problem is that some of them are incompatible with SDS and some surfactants. So you have to read about those protocols, because they will give you results which are not actually true. And for instance, for BCA method, you will have to measure, as you can see, at certain wavelengths. And since it's a colorimetric method, what do you think? Will the change in this color be linearly proportional to the change of concentration? Have you had any laboratory work on spectrophotometry? So what is the rule to get concentration if you have some absorbance? Let's then try to draw it. Like here we have some absorbance here, for instance, and some concentration of our, let's say, it would be a milligram, for instance. Will it be something like this?
But maybe it should be logarithmic.
Yeah, absolutely. Why? Let's say we have one cuvette of some known concentration, and this cuvette will allow us to pass only 50% of light at given wavelengths, right? Let's say that we have the same cuvette here, and it will also provide us only with 50% of light, but it's already 50, 25. Then another one, 12.5, and so on, so on, so on. Right? What is the problem here? Even if you have double the concentration here, the actual transmittance will vary less, right? And if you have change in concentration and a slight change in concentration here, you have 50% to work with, right? So it will be something like logarithmic, you were right. If concentration is small, it will be something like here. And you will have to have at least 2 points to calibrate this curve. Actually, 3 is more useful to be able to get from these numbers to real concentration. As you can see here, there is one of series of wells which actually have change in color, right? This is calibration for your physical. You just put some known protein like albumin, for instance, and first one is no albumin, another one is, let's say, 1 microgram, 10 microgram, 100 microgram, and so on, so on, so on. You can calibrate them and get an answer of what actually your concentration is. But the problem is that we need at least 2 or better 3 points, and if we want to calibrate really cold display, we'll need a little bit more. Of course, there is different. Like Bradford is the same as here, but we already have some problems. First problem is that we need a lot of points to calibrate, and another problem is that most of those assays will not work with some other chemicals which present in our samples. And there is one solution to this. There are new fluorescent dyes which binds to protein in quantitative manner, and you can use them actually to get the concentration. Most of them have covalent bond to lysines, and if your protein is long enough, there will be some lysines there. And actually, for proteins, you know how much lysines will be in, on average, in this protein. And it can bind to them in, when it is in native state, I mean this fluorochrome, it will not fluoresce at all. And only after the covalent bond, it is covalently bind to the protein, it will start to fluoresce. What is actually fine here, as you can see, this calibration curve and standard is linear. So for linear function, you actually need how many points? 2 points only. And also, most of them, as you can see, tolerate salts, solvents, mercaptolol, free amino acids, which can be a problem actually if you are working with cell cultures, because if you do not wash your cells well from medium contains those free amino acids, right? DNA contamination, reducing agents, and so on, so on, so on. So you can just look at this table, get whatever fluorescent dye you need, calibrate on 2 points, of course more points are preferable, and get this concentration. It's easier than all those homogenic assays. So now with no concentration, we can turn the quality of our separation by using just spectrophotometry, because for aromatic amino acids we'll have this absorption at 280 nanometers. Usually you try to avoid this in work with nucleic acids, right? But with proteins, which actually are proteins, we want to use it. But what do you think? Why we are not using just absorption is this wavelengths to get the concentration of our proteins? There was a lot of advertisements of such an instrument which was called nanodrop, if I'm not mistaken. They were pretending to measure everything in one run, and they say that we can measure concentration of proteins without any problems, but actually results were a little bit strange. So why do you think it is a problem?
Because of these aromatic groups.
Yeah, no, we have not so much of those nucleic of those amino acids in our proteins, and some proteins have a lot of them, some proteins have less of them. It's not the lysin which is only present. Also, other chemicals can absorb in these wavelengths, so we can't account for it. And our results will depend on SDS concentration, on phenol concentration, if some phenol is here also. So that's why we need those specialized assays or fluorescent dyes. So now we have concentration, and the last thing that we need to check is whether a protein is in good shape or whether it was eaten up by proteases. We need to make some gel electrophoresis and then just to see whether it is okay or not. So, what I want to demonstrate is some bad results. Actually, this electrophoresis is from human tissue that was embedded in paraffin for more than 20 years. And actually, it is strange, but one of protocols can get some proteins from even such old tissue, which was at room temperature and so on. Other protocols were not possible to do so, but you have a lot of degraded proteins here. It's how it can look if you have more or less good sample and some samples which were degraded. Of course, it's some proteins which are highly present here, since it was a nervous tissue. Mostly it's cytoskeletal proteins, but still. So, now, we want to start to study a proteome, and first we have to discuss SDS page method. Was it already, have you done SDS page by yourself, or was there in your body of work? Okay.
Oh, I did it once.
Once. Okay. So I think I need to explain it a little bit at least. Now we have our proteins which are coated with SDS, and actually we can start to use them for electrophoresis. But there is though one problem. The problem is that if we'll have electrophoresis and if we'll start to work with it right on the start, we'll have a problem of diffusion. Let's say we have some gel, some wells here, and since we have more than one, we'll start filling it one by one. At the time we fill the last one, in the first one, our protein will already be filling almost the whole well here, right? And if we'll start continuous electrophoresis without any special measures we will be talking about, we'll have, even if we have only one protein in mixture, we'll have such a big band here, right? It's not what we want. What we want actually is to be able to slowly fill all the wells, be able to focus all the proteins in as small as possible line, and if it will be possible, then if we will be able to somehow compress all the protein with a thin band, then this one protein will have only one bond and it will be only compressed. Also, if you have multiple proteins, it's even worse. They will be covering one another. But if we can somehow compress them, we'll be able to resolve them easily. And actually, that is what's so interesting about this classical lamellar system of SDS page electrophoresis, because we will use so-called discontinuous electrophoresis. We'll use different ions there, and we'll make sure that our protein, before starting the separation itself, we'll have different gels here, will be compressed in this thin line, and only after this the separation will begin. Since we have 8 minutes before the break, I think that we'll have a break now and then continue with SDS page and how to properly do it. So 10 minutes break.
Чем хороший? Ну чего ты? Меня скидывают больше, чем выбирают. Я теперь и видимо, я буду.
Коллеги, знаете, выключается.
А перестаю, да.
I'm sorry, but it was just a bit boring. So it was. Спасибо огромное. Alright, let's continue. So the problem with the aphoresis was that we had somehow to focus proteins to a small thin line before we start our separation. And here comes the main idea about this SDS page is to use different buffers. For instance, we will use water-soluble amino acid glycine in cathode and anode buffer, but in the GAL we'll have a different pair of ions. We'll have a trace, it's the same one, but we also have chloride here. So we have different charge carriers. For if you will have no charge carriers and apply some voltage, no current will flow and nothing will happen. And here we have different charge carriers, And this glycine is quite interesting because, as all other free amino acid, it can be in different form. The form is positive, negative charge, and twittonic form where it is almost neutral. And as you can see, we'll be also using two different gels, so-called stacking gel and resolving gel. Stacking gel will have, as you can see here, different pH, almost normal 6.8, at which this glycine will be in twittonic form without any charge. And what will happen when we start our electrophoresis is the following. Let's see that we have a lot of glycine here. We have chloride here and here, and we also have a glycine here. I'm not using here 3 because it's the same throughout the whole gel, but when we start to apply our current here, those charge will start to move, and since glycine have not that charge at given pH in this stacking buffer, there will be no charge carriers because chloride is small and it easily migrate through this gel. Just have, it will be migrating here, and this gel will have only glycine, which at this pH will have no net charge. What we have here? We have a lot of charge carriers here and here, but this area have no charge carriers except for what we put here? Proteins, right? So it's the same as we have only proteins in our solution, right? And if we'll just draw some electric scheme here, we'll have some very small resistance here. Of course, it will be ballpark, let's say some 10 ohms. Of course, it's not so, but just we have a small resistance here, let's say 20 ohms. And we have a lot of resistance here because only proteins can transfer charges here. Let's say 10 kilo ohms. Since we have, let's say, 100 volts here, we can see that due to low resistances there, we'll have a high high strength of electric field just in this region. Almost every of this 100 volts will be there, right? Because those resistances are low enough, and we have here a lot of resistance. And it will make our proteins to really fast go there. And this interline between stacking and resolving gel will be the line where all the proteins will just stop. Because they can't move by themselves, they need some charge carriers to push them through, but nothing here yet. And our glycine here is also without any charge. So it will just be there. And then glycine will also migrate. And this pH will be different and will have charge carriers. And this glycine will push our proteins through resolving gel here, but from one starting position. That's actually what was the main idea, and Nobel Prize for electrophoresis was mostly also because it wasn't in gel, it actually was in solution and so on and so on, but the idea of being able to do discontinuous, we have different ions and we have different gels here, discontinuous system allows us to focus on our proteins in this thin line, right? Actually, we can use other charge carriers aside from glycine, because glycine for us is not quite usable for small proteins, less than 10 kilodalton. Why? Because we have a lot of SDS in our page, in our solution, in our samples, and this SDH is actually salt. And if you have a lot of salt, it will form micelles. And those micelles will hamper the migration of our protein, small proteins. It will be just all in one line. And if you run such a gel, then you will have all your 10 and less than 10 kilodalton proteins in one line. There will be no resolution for such a small proteins. And we can use actually different charge carriers aside from glycine, such as tricine is also possible, and other systems which can solve this problem, but will lose resolution for big proteins. So it's either you work with small range proteins or high range proteins. Was it understandable? Are there any questions about this idea of focusing your protein before resolving it in a resolving gel? What do we actually need for this to happen? Can we use just one ion, be it chloride or glycine, tricine, whatever? It will be a continuous system, right? And all the proteins will start from their position as here will have the same. Also, can we use some charge carrier which will have the charge at every pH? So we have to have some charge carrier which is CTO in form and can allow us to have a high change of field in this small region to focus our samples. And also we can have different gels. For instance, you can use gel with, which have big nets, and all the proteins will migrate through it easily. And you can change this gel here to have smaller mesh size to resolve our proteins there easier. The idea behind this way of resolving proteins is the following. For instance, we have a lot of people who have to run through the forest. Like there is a forest, a lot of trees. For one runner, you will give this stick. For other runner, you will give broomstick. For some other runner, unhappy one, you will give stick that is used for jumping at Olympic Games of this sport. And you ask them to run as fast as they can through this forest. Idea is obvious. The one with the chopsticks or with cranium will be the fastest one. But what do you think? Will the results be always the same if you will do the same electrophoretic experiments from day to day? Is it possible that something will change?
Yes, temperature maybe.
Temperature, right. That's okay. Also, some salts in your buffer can be different, and so on, so on. Gel itself can be different. So you can't actually get molecular weight without some standard. You have to use protein ladder. You have to add, at least in some of your wells, some known standards of known molecular weights, which will be resolved aside from your proteins. And then you can just say, this one is 200 kilodaltons. Then these proteins are between 200 and 150, right? So, unfortunately, we have to use them. But, I have a question. What do you think? Will there be the actual molecular weight of this protein available for us just by comparing them? Or will it be something different?
Something different,
Like, is it possible that we have a protein which is, let's say, 150 daltons, but it can be seen on this gel as 140 daltons, or a different condition. Let's say we use not glycine but some other ion, like 160 Dalton or so on. And what can lead to such problems?
The jet goes become much smaller, much larger.
And it's possible, but also we assume that SDS covers our protein homogeneously, right? But what it is protein have some part of which is highly hydrophobic. SDS will preferentially bind to this part of protein, right? And will not have this one-to-one ratio between size of our proteins and number of SDS molecules. And this ratio can be modified by buffers, the kind of gel we used, and other ways. So it is not possible to say that this protein have this molecular weight without using some bioinformatic approaches or mass spectrometry. We can see that it seems to have this weight, right? Because we have different systems of electrophoresis, and we can get different results. But usually, if you have the same system, for instance, the trees license system with the same gel, will get the same order of your proteins. So the order of them will be the same in this system. In other system, it could be a little bit changed, but still. So let's then continue, and I want to show you some small program which is freely available. It's called Gel Analyzer. Of course, there is a lot of programs for analyzing your gels. Some of them are commercially available, unfortunately not available for us right now. And this program is free to use, absolutely. And we can actually use it to analyze gels. Let's just have some gel here. First, our program asks whether it's light blobs on dark background for fluorescent gels or vice versa. We have dark spots on white background. We can rotate it, shear it, whatever, or cut some part of it, but it's okay. We can crop it, but we don't need to crop it, right? And here we can start to analyze it. We can ask program to do all the hard work for us, or we can manually actually put all the lanes here, mark all the parts, but let's start from automatic work. First of all, we ask program to find the lanes of our gel. As you can see, it detected pretty well without any problem. We don't need any corrections, except for this one, I think, but right. Then, we want to detect bands inside our lanes. We can also do it automatically. Here are some parameters. I will just change them a little bit, which you can change in order to detect all of your bands. Let's see if it works right or not. Well, mostly it does work, but not quite well. Let's change it a little bit. 21, 5 for instance. It's a little bit better, but we lose some sensitivity. So you have to play with those parameters if you want to do it automatically. But you can do it by hand. It is possible to just put somewhere, your mouse somewhere, and you will add additional bands there. Okay, now we have bands. We have we can actually see the densitometer here, and you can change if something is wrong, like here. And what we have here, the problem that is still remains, is that there is a base lane. Even if there is no protein, we still read some numbers, and we want to make this foreground non-existent, and we can actually start it by another program here, and we can automatically subtract background with different algorithm. I think that line ball is the most usable here. Now we have actual data, and we can count the number of pixels in each this band to know how much protein in this band, right? Program can do it for us, no problem. It's already actually done it, as you can see. And we are missing only two things. One, we need to calibrate molecular weights. And if we have some dilutions of our samples, we can calibrate for it. If we have some known standard of proteins, we can calibrate even for the quantity of protein here. But let me show another usable thing is that we can actually, if our gel is not as straight but a little bit bent, it's possible for this program to add the correction for this bending of gel. Just click here and put points for the same molecular mass, and it will correct the whole gel for you. You can add 3 to 5 different lanes for correction, and it will be corrected. For this gel, I don't see any need for correction, unfortunately, so yeah, it's fine as it is. And now we can make molecular weight calibration. Here I have a standard of known weight. It's 200 kilodalton, 150 kilodalton. Oh, sorry, I was wrong. And it already starts to fit it in some model, and as you can see, for 3 points we can actually get this nice idea, but let's continue. Probably it wouldn't be so good afterwards. It's It's a tracing system, so it could resolve less than 10 kilodalton. And it is non-linear gel, so, well, yeah, it's not quite good, but still 0.97 can work with it. So, now we can click on any other plane, and we have all our bands already calibrated using those molecular weights we get from our standard. So, pretty easy, right?
Mm-hmm.
Program is freely available. Just go to this gel analyzer website, you can play with it, it's free. Also, you could export those data for in any spreadsheet, and you can calibrate even for quantity of protein. I don't know what was the quantity of the protein, so it will be some ballpark, but let's say we have some 10 microgram, let's say 10, let's say 20, let's say 30 for instance. What is nice that it also tries to get some model for it, and of course it's not 20, it should be a little bit more, for instance. We can try to make it better. And after this calibration, you will also be able to get a volume in units that you use for calibration for your gel. So, as you can see, you can use this program to get molecular weights of your electrophoresis gel, and if you have standard for quantity of the protein, you can also use program for quantitative work. Alright, then let's continue then. Now we know how to run the gel, now we know how to what to do with the data, with the picture, which you can get even with a cell phone. You don't need actually this gel imager for routine gels. Now we will have to talk about how we actually can make those gels by ourselves. Of course, you can buy already pre-casted gels, no problem. But nowadays it's a little bit costly and a little bit troublesome to get them. And actually, you can cast those gels by yourself. Only 4 chemicals that you need. Actually, 5. You need a buffer also. You need acrylamide, bisacrylamide, and something that provides free radicals for the polymerization. Here we'll have APS as a provider of free radicals and TMAT as a catalyst for APC to provide them. Why we need different chemicals here? Acrylamide itself will polymerize in a linear polymer. And linear polymer is not quite usable in our case because we want to have some meshes in it, right? And if we'll add acrylamide and bisacrylamide, as you can see, bisacrylamide have 2 groups that can be used for polymerization, like here. You can use different concentration of those chemicals to get different gels with different pore size, with different stiffness, and so on and so on. So you can cast different percentage of acrylamide to get gels which would allow you to resolve protein in different range. For instance, 7% will give you this range, 15% will give you this range, except for, as we already discussed, that glycine will not allow you to resolve 10 kilodalton and less. And also you can cast those gels and put the same set of proteins to see what position will they occupy in different percentage. And as you can see, when percentage is low, you get good resolution at proteins with high molecular weights. If the percentage is really high, you have more or less good resolution at some lighter proteins and peptides, but all your heavy proteins will be almost non-resolvable. You can get some medium percentage where you can resolve most of them, but as you can see, you lost some of them during electrophoresis. And what also can be done, you can make so-called gradient gels, where this percentage differs from the start of your resolving gel to the bottom of your resolving gel. And it will allow you to simultaneously be able to get more or less okay resolution of high molecular weight proteins and of low molecular weight proteins in the same gel. So actually you can cast such gel by yourself. It's either you need this equipment, just 2 vessels which have some stop caught between them, and a vortex mixture with magnetic stir. And then when you put 2 different concentrations of your gel here and here, since you put the most concentrational solution here, it will start to mix. And first you will get high concentration of acrylamide, then you lighter solution will be mixing with your heavier solution, you will get some smaller concentration, and at the end we'll have only the solution here, and we will have smaller concentration of acrylamide, and you will be able to cast such kind of gels. If you do not have this kind of apparatus, you can do it even with a pipette. I will just try to open a YouTube video.
Action.
And
Let it go all the way up.
that's how it can
And
be
then
done.
you mix it with
Just
inserting
one small
single bubble
here
into the pipette. Let
will
it go
mix
all the way up.
2 different sets of your
And then
acrylamide.
very
You
slowly
can just cast it down.
pour the
So
gel
you
in.
can get 10 ml of, let's say, 20% acrylamide, 10 ml of 7%
This should be the
acrylamide,
rate.
just 1 bubble in here and cast gel like
It's easier
this.
to slide it
You
along
have
the
pretty
glass.
Of course, it wouldn't be as good as precasted ones, but they would be a lot cheaper, and it's not a big deal to make them by yourself. So, if you'll need to have a good resolution at both sides, at heavy and light protein, you can do it by yourself even without special equipment. So, let's continue. I've already mentioned that there is a problem with small molecular weight proteins and peptides in our tris glycine electrophoretic apparatus because they will look something like this. No way to resolve them. But we can actually get different system, this tris system for instance, and with this system we can use different molecules instead of glycine, like MES, MOPS or tricine, and it will allow us to resolve even those small proteins. As you can see, the difference are obvious, and neutral pH for this is also good because proteins actually at highly basic conditions, which we use with our glycine basic gels, can be modified chemically just because of those house conditions, because of SDS and so on. With neutral pH, these trees will have a little bit different pH for zwitterionic form. Will have less of those spontaneous modifications, and as you can see, these gels look a little bit clearer, in fact. So, not only glycine is used, but also other forms, and you can use neutral gels as well. Well, we can skip this. We can skip this, and we can compare different systems between them with the same set of proteins. Some proteins are internally colored just because of primary and secondary structure, in this case primary structure, and we can see them during the electrophoresis on those pre-stained letters. And first three are tris glycine with different percentage of acrylamide and different percentage of um, um, gradient gels. The other is this tree system with MES and MOPS, and other is tricine and tris acetate systems. As you can see, they will provide you with almost the same result, but resolution in different protein sizes will be different. So if you want just to see what's inside your samples or make some quality control on gel, it's not a problem. You can get 14% gel, for instance, and get some results. But if you are more interested in some higher molecular weight proteins, you have to either get those um, gradient gels or use, let's say, MOPS system or even acetate or tracing system. The differences are obvious and you have to experiment with your samples actually. From my point of view, neutral bis trees system is better. If you have money and abilities to use them, they actually provide you with better results. So, what do you think? Are there any cases when we would like to have denaturated protein electrophoresis without any denaturation of our proteins, I mean? With this electrophoresis, with this DSPH, we have to denature our proteins, right? Are there any cases when we would like to separate our proteins without denaturing them first?
Yes, maybe purification of this protein.
Absolutely, because SDS is not as easy to get rid of like urea, for instance, right? And also, you have complexes of proteins, right? If you denature them, you have just monomers here. And sometimes you have interest in those complexes. What monomers are here? How many of them? And so on, so on, so on. And if you will start with denaturing electrophoresis, you will lose this information. But if you will start this electrophoresis with native proteins, then you will get some information which will not actually give you isoelectric information on this protein and not molecular weight, but something in between, right? And also, some proteins will have positive net charge, some will have negative net charge, and what phrases will you use in this case? Because either case you will lose half of your proteins, right? But there is a solution for this, and you can use some dyes like Kumasi blue, for instance. Kumasi blue is actually a dye that we use to stain our gels, and Kumasi blue have ability to stick to proteins. And what is really nice, it have small negative charge. And if you have this solution of this dye at some small concentration, it will stick to our protein, add some charge, but Kumasi blue is not SDS, it will not denature our proteins. So we have a protein which is coated with some negative charge, which is possible to use for electrophoresis, but it will stay, this we hope, it will stay in its native form. And even if protein have net positive charge, Kumasi blue molecules would be prevailing, be a lot more of them, and even our positively charged protein will, after this, have negative charge. And we can use just one polarity for our electrophoresis to get all the proteins. If something is not understandable, just ask. I do understand that gel electrophoresis is a little bit boring, unfortunately, but we have to cover this topic. So, how will this kind of electrophoresis look? Let's say that we have some complexes of our proteins here, and we go to do those blue native page, which is our protein with some Kumasi blue, which is used for electrophoresis. We'll have those not quite resolved bands here, which represent different complexes in different ways. What we can actually do next, we can make second dimension. We can take this part of our gel, orient it this way, put it into stacking gel, and now make SDS page electrophoresis. And we'll have something like this. Each complex is here, and here is a monomer, which were in each complex. Right?
Is it a good picture of such?
Yeah. It is the best picture I was able to find in some publication. Actually, mostly it looks a little bit worse than this. But still, it's only method for us so far. We haven't been covering any other methods for protein-protein interactions to see them. Like, if you know that it was some complex, you see that there are 1, 2, 3, 4, 5, 6, 7 different proteins, right? At least. And actually, you can make it a little bit differently. You can use something that is not so strongly denaturing as SDS. SDS is, of course, quite denaturing agent. And you have monomers, you have everything is in this repair. You can use some less denatured substances like the dicyl maltose, for instance, and get pictures like this one. Resolution, actually, it's a really good resolution for such kind of electrophoresis. But even from this electrophoresis, you can just cut a piece of your gel, and as you will see the next lecture, you can identify the proteins in this band. It's quite enough to excise even those two bands as a separate ones. So, if we can get our results, why do anything more? It's possible. So, now we have not only a way to see monomers of our proteins, but we can even work with protein complexes either strongly denaturing them, like here, and getting monomers, or having some slightly denaturating way to just get the same complexes. It's a mitochondrial proteins. You know there's a lot of complexes of proteins in mitochondria, and you can see that in first separation it was absolutely native, blue native gel, and in this separation you use gel with some addition of slightly denaturative maltose here. And each component of this complex is visible, but probably some of them are actually a lot of monomers which are somehow connected. We use only mildly denaturing agent, so we can compare this and get some information from those two gels as well. So, the last one, the last system from electrophoresis that we also need to cover is acid urea electrophoresis, or AUPH. Sometimes you have to work with proteins which have highly basic nature. There's a lot of amino acids that get them some charge, and we have those basic proteins, like, for instance, if you're working with histones. It is a problem for native blue native electrophoresis, but we can use such conditions as using some acid and urea for electrophoresis to get such pictures. Of course, it's not quite good resolution, as you can see here, right?
Mm-hmm.
But you can resolve those proteins, and also if you want to do some preparative work, it's easier to get rid of urea than of SDS. SDS is really hard to get rid of. It's like a soap; it's always there. But with urea, you can just change buffer, and you will have your protein without this urea. And actually, your protein can even participate without it, so you can even clean it. And also, there is such a technique as using, again, 2D gel, using acid urea with some Triton 100 in the first dimension, and SDS page in the second dimension. This way you can get information about molecular weight, SDS page can provide you with it, and you get some information about proteoforms, because acid urea and Triton page allows you to separate different isoforms of the same protein. For instance, you can probably know that histones are proteins which are highly modified, acetylation and so on, so on, so on. And those modification changes protein and its position during this phoresis. So you can actually get some information just using electrophoresis, which can be done in any laboratory. You don't need any mass spectrometry equipment for this. And afterward, you can excise this part of gel and get amplification. So, the electrophoresis apparatus, which we use, there's a lot of different types of those apparatus, but what we need here is to use special sample buffer for SDS page. We've already said that we can actually focus our proteins in a well. That's fine. But we still have some problems. If we'll have a lot of protein, what will happen? Will there be enough SDS to cover a lot of proteins with such a small volume? And if not, proteins will stick to each other, and we'll have something that will not move to the gel, right? So we need to add some additional SDS here. Also, if we'll use only water, we can have a lot of time pouring our samples in this lens, and if it is in water, it will diffuse out of this well. So we need something that makes the diffusion smaller, like glycerol. Also, glycerol is very beneficial because it's heavier than water, and it will actually put your sample here, down here, and just for the ease of use, it would be nice to have something that have color, because proteins mostly uncolored. We can use some broken blue, for instance, just to be able to see how we put our protein in these wells and whether there are some cracks in gel or something like this. And also we can see the rise during run, because this dye also have some charge, including migrating in this front, and when this front will be present while gel, electrophoresis could be considered as down, we can stop it, right? So that's why we have those components in a sample buffer. We need glycerol to make it heavy and less prone to solution. We have additional SDS, we have some trees as a buffer, we have bronphenol blue as a color, and also we need to take into consideration that there is a lot of cysteine bridges in our proteins. Will it be possible to denaturate such protein just using only SDS? No. That's why we need some agents which can actually break those cysteine bonds to actually linearize our protein. Because if it wouldn't be a case, then we'll have two different bands for the same proteins. One for proteins which have all those cysteine branch intact, and one which have lose some of them by some chance. Now we can use some chemicals to disrupt all those bonds, and we'll have just a nice version of our protein. So, also I would like to give some practical example. For instance, we get our proteins, we use our lysis buffer, we boil them, so we hope that they are already denatured, and then we have some break in our work. Let's say we have our Saturdays and Sundays, and we put our sample in, let's say, fridge, for instance. What will happen with our proteins during this time? Is it possible that there would be some spontaneous renaturation of proteins in these conditions?
Actually, they can.
They can, yeah. We've already talked about those highly hydrophobic parts of proteins. And if you will keep your sample after lysine buffer for a long time and then start to do SDS page electrophoresis, you will have some problem. Some protein will have one bond, some proteins will have multiple bonds because this renaturation. And practical thing, you have to boil it again before you put it on your SDS page analysis, just to make sure that everything is actually denatured. What you can change here is time and voltage for your gels. Of course, we want to separate our gels as fast as possible, but there are two problems. First problem, the more the voltage here, the more the current. The more the current, then the more the heat that will be produced. And if we heat our gel, we'll actually make the separation different, and if we heat it even more, we'll have participation of protein inside our gel. So faster is not always better. Start with, let's have this small kind of mini gel, 100 volts would be enough. Even 80 volts can be enough. And if your voltage is too high, you will see such strange picture. All your lanes will be smiling at you, just to say that your voltage is a little bit high. Also, you can make your gel to run at lower voltage and higher time. But there is also a drawback. For instance, you want to have fine gel and resolve it at, let's say, 20 volts for one day, but there is some diffusion. And you will also lose resolution just because diffusion is comparable to the speed of moving of your molecules. So don't try to get those extremities too fast or too slow. Somewhere in between is better. So that's some practical news. And I've shamelessly stolen some of illustration from this really nice blog. I really recommend you to go there. It's blogged by some biochemistry also has a really nice channel on YouTube where she demonstrates all those methods we are covering actually, so you can see how it's done by hand. So shameless to stolen again. If you have our gel after electrophoresis, what do you think? Will it stay the same after some time? Or what would happen with our proteins? Will they stay there, or will they try to diffuse? Diffuse, right. So if we want to stain our gel, we first have somehow to fix our proteins inside the gel, right? To make them insoluble, for instance. And afterwards we can stain our gel and have our picture. We can even dry our gel and have it just on a paper and put it in some thesis or whatever. So we have our gel, and then we have to stop everything that is happening in this gel. How we can do this? We can add acid and alcohol. We can make different pH. It's basic pH, we make it acidic pH, and we get rid of water. What will happen with proteins in this environment? There is not enough water, and the pH is now acidic. It will try to rehydrate, right? It will try to stick, and it will actually participate inside the gel. That's what we want. It won't move anywhere from those spots. But if you want to get this protein from the gel afterwards, it will be impossible. So we can't actually stain the gel or stop the gel this way and then have some blotting experiment, right? Because this gel will be already participated inside the gel. It wouldn't be possible to get it. But for mass spectrometry, we'll be able to get this protein just by excising it with proteases. So actually, you can stain your gel, you can excise some parts of your gel for fusel analysis for identification of proteins. So we've done all electrophoresis, we stop all the possible movement of our protein. Now we can stain it. Actually, there's a lot of different stains we can use, and most common one is Kumasi. But from practical point of view, I strongly suggest you to use so-called colloid Kumasi. Colloid Kumasi is easier to use and more compatible with mass spectrometry. It costs about the same. So if you have choice, usually it's better to use so-called colloidal Kumasi. What we do, we get an excess of the stain, and this dye will stain the whole gel. Not only your proteins, but it will be inside it. So it will be just blue, dark blue. And then, as we usually do in histology, we try to so-called differentiation. We try to get this dye out of our gel, and only the places where this dye have something to connect to, like proteins, it will stay there. But all the other will be washed away. So, after staining, we have to do destaining, which is usually done with methanol and glycol acetic acid. And there is also some tip from the laboratory. Methanol is costly, acetic acid is a precursor, so we have to somehow conserve them and use them as less as possible. It's possible to destain this solution, because all the dye which we will use for destaining our gel will end up in this solution, right? If you add to this solution either Kim wipes, those paper sheets, or if you use some foam rubber, it have the same groups as our proteins, and this dye will stick to it, and your solution will be colorless again. I don't know whether whether this foam rubber is well, it has turns out in Russian actually. Prolong, if I'm not mistaken. This yellowish thing which is used actually to pack things. It has the same growth as proteins. And if you try to distain your gels and put some of this foam rubber in the solution, the solution will take some dye from your gel, and this dye will try to stick to this foam rubber. And you can distain it more efficiently and without using a lot of chemicals. So just a small tip to use in laboratory. And after that, we already seen how to deal with our gel. We need to photograph them, we need to use this program, and we can calculate whatever we want from it. Also, as I've already said, there's a lot of different ways to stain your gel. You're not limited with this Kumasi. Kumasi is really nice because it's easy to do, but we have specialized dyes. I actually prefer fluorescent dyes, and the pity it's not possible to photograph such, to get pictures of such gels with just cell phone. You need special illumination system and some additional filters for it. But I really like those fluorescent colors because you can adjust sensitivity by using different exposure. You can expose it for 1/100th of a second, 1 second, 10 seconds, and you can see really small signals just by using long exposures, up to some, well, more than 100 times more than you can do just with some small exposure. And also, there are some special dyes which can stain you only proteins with some modifications. Unfortunately, only one such dye is available commercially, you can buy it. Other dyes are published in some articles, but you either have to ask authors to get it or to synthesize it by some chemists. But there are some special fluorescent dyes which can bind to glycoproteins. This one is actually commercial. It's ProQ Blue or ProQ Diamond. Yeah, it's just a little bit different color. And you can stain your gel twice. You can stain it first with this ProQ Diamond stain and only see proteins which are phosphorylated. And then you can add different stain. You don't need to distain the gel, only places where these proteins or modification will be fluorescent. And stain it again. And now you have the whole picture of your protein, and you know which of them are actually phosphorylated. Clever idea, right? Unfortunately, only one dye is commercially available. All the others are published, but yeah. Actually, there's a lot of different dyes. I won't be getting into it. The only one that I want to talk about is this silver dye. Because sometimes you strive to see really small quantity of protein. Like you can't see it even with fluorescent dye. It's impossible to detect. And this silver dye is actually something that works the same way as black and white photography long ago was. We have some salts of silver, and those salts can be easily reduced by the proteins that are in a gel. And what will you have afterwards is a small crystal of silver in your protein. And this crystal by itself, with the use of special chemicals, can be used to grow even more crystals on it. So it's some amplification of your signal. And you can stain your gels using some salts of silver to have really nice amplification. But there is a drawback. It's impossible to use such gels for, let's say, proteomic analysis. It's impossible to use it for mass spectrometry. I know that there are some publications that says that it is possible to, you just have to try this, but in my hands it doesn't work. I don't know when, probably my hands are a little bit curved, but unfortunately it's really nice for detection. And there's some companies like Bio which use some chemical to modify proteins and to be possible to visualize it in ultraviolet light. Don't know whether it's actually have any additional value, but it's also possible. So we know what to do with the gels, and also sometimes we want to detect not all proteins that are present in our sample, but some certain proteins, and we can use antibodies against this protein to see this protein. And as we've already talked about gel electrophoresis, we know that we can't fix our gel because if we fix it, we won't be able to transfer our protein to anything else. What do you think? Why we can't do immunodetection inside gel? Why we need to transfer our proteins to some membrane?
Because immune detection, it uses antibodies
Yes.
and maybe it's not connected.
It is connected. Actually, first of all,
Antibodies always, not maybe always, but often they are, how to say,
you can penetrate inside gel. I mean,
um,
can get inside the gel, right? Even
yes,
this.
but they should be included on some surface to.
They can be used in a solution as well.
Oh, okay, maybe.
Actually, it's quite easy. First of all, our gel is about 1 millimeter, for instance, and our protein is diffused in this gel, right? If we just pour antibodies here, even if they can't see some proteins, they'll be able to see only proteins which are close to those borders, right? And it would be just some part of concentration. Also, those proteins wouldn't be fully exposed to antibodies. What we want something on top of which our proteins are just lying and they are present into antibodies. That's the main reasons why we want to transfer our proteins from gel to some membrane. Because if it doesn't penetrate inside the membrane, then it's all available for antibody. We can probe any part of this protein, right? So that's an idea why we need this blotting. Since we have our gel without fixation, we can use either electric current, capillary forces, or vacuum to transfer our proteins from the gel to some membrane. Long ago there was a lot of different materials for membranes, but nowadays only two types are used. It's nitrocellulose and PVDF. PVDF is really good membrane, but there's one drawback. It is highly hydrophobic. And we have our proteins in solution, right? So if you use PVDF membrane, you need to make it hydrophilic somehow. You can do it by soaking it in methanol, 100% methanol, and then putting it into water. Then all the hydrophobic parts will be coated by some methanol, and they will be hydrophilic, even if it will be stored for a long time in water afterwards. But it will be possible for proteins to stick to this membrane because of those hydrophilic interactions. And nitrocellulose is actually a kind of almost paper, though it's really flammable, so don't try to put any fires on it. And nitrocellulose is hydrophilic, you don't need any methanol to make it work. So, for PVDF membrane, we need this pre-treatment. For nitrocellulose, it's not needed. It doesn't mean that something will happen, but just we don't need some additional treatment for nitrocellulose. And we have to put such a sandwich of different components to make this transfer run. We need some sponge which have some buffer in it. We need some filter paper to have our sandwich of membrane gel between them. And this paper also helps us to get rid of any air bubbles because if there will be any air bubbles, current will not be flowing through it, and no protein under this bubble will be transferred. So we have to get some roller to just get rid of all the air bubbles trapped here. And since our protein is still covered with SDS, it has this negative charge, and we have to be really careful to put membrane closer to anode. So our proteins will be moving towards the membrane. Unfortunately, with such kind of electroblotting, there is a problem. The problem is following. If we have really small proteins and really high current, they could migrate through our thin membrane and be lost in solution. If we have too small current, then our big proteins will not be able to cross this border to get from the gel to the membrane. And we can get some optimal voltage which allows to, at the same time, have heavy molecular weights, low molecular weights protein on the membrane. We have to improvise. We have to try some different voltages or voltage steps or something to be able to transfer as much of protein as it's possible from gel to membrane. But there is no one condition which allows you to transfer all different proteins to the same efficiency. So, afterwards, we can use different apparatus which can do this electroblotting. As I've already mentioned, we can also use capillary effect and vacuum to do the blotting, but most of the time we use electroblotting with SPH gels. And there are different types of such instruments. The most usable one is semi-dry transferring system. We have to make this pile of filter paper, gel, membrane, filter paper, put it in some apparatus, use some current here to transfer your proteins, and it's possible to get membrane with proteins. It's also possible to use such devices where it is easier to do, because there are some springs which compresses your gel and blot and allows you to do it in, let's say, 7 minutes. Just 7 minutes can be enough for most proteins, but it's costly. And also you can use so-called wet electroblotting, where you can put both your membrane and your gel inside the same the same tank as you have been using for SDS page, and it will take a lot more time, but it will get you more protein blotted to your membrane. So it's either fast but not complete, or long, but unfortunately it will take a lot of time. By the way, do you see any problem on these pictures? Because it's a picture from a textbook and there is one error on this picture.
I'm not tested.
Yeah, it's a ketone, but it's a red, and it's anion, but it's blue. Why? Don't know, but it's in it's from textbook, and still. So let's continue. What we do, we have our gel electrophoresis. We can transfer it to our nitrocellulose membrane. We actually can stain our proteins on the membrane, but using different stain. Here's a recipe for this solution. I don't see actually the reason why we need to stain our membrane, except for being able to be sure that most of our proteins were actually transferred to our membrane. But mostly you just stain your gel afterwards and see what is left on a gel. It's more than enough. Afterwards we can do immunoblotting. We will be discussing it a little bit later. So actually you can, if you want, stain with some copper reversible stain your gel, but why? I don't see the reason. So usually you make this transfer of some gel which you already analyzed by just SDS page in massive stain, and you know what is in your gel. So now we have a membrane, either nitrocellulose or PVDF, and I have to remember that for PVDF membrane you have to use methanol, otherwise no protein will be possible to bind to this membrane. With nitrocellulose it's not necessary. And we can actually stain the gel after transfer. And as you can see, what I've mentioned, there are systems which allows you to make this transfer pretty fast, under 10 minutes, even 7 minutes for some systems. And with wet tank transfer, it takes hours, but as you can see, the efficiency of such transfer for wet tank is higher. So if you work with really small amount of protein, wet tank is preferable. But most of the time, from my experience, those machines which allow you to use 7 to 10 minutes are more than enough. So it's only some rare cases when you can use this. And we can also stain membrane, as you can see. There are differences, of course, but mostly it is transferred without any problem. So we have our membrane, and now we have to discuss a little bit about antibodies. Have you had any courses on immunology, or so you know everything about antibodies, what type of antibodies are there, and so on and so on. Mostly with Western blot we use IgG antibodies or IgE antibodies because of their structure. Also sometimes we use antibodies which are small enough, which are either from camels, they have only this FC fragment with some addition, or we can actually get fragment from antibody by proteolytic activity. But most of the time you will have to use IgG or IgE antibodies. Also, we can have either monoclonic or polyclonal antibodies. I think it's known that polyclonal antibodies will be able to recognize different epitopes on the same antigen. So there will be different places for recognition of your protein. But if you are working with proteins, what do you think? Can it be that a lot of protein can share the same motif?
A lot of protein share.
A lot of proteins, yeah, we call domains, right? So if I have some antibody, polyclonal antibodies, and some of them recognize this domain, they will recognize not only our protein, but also some other proteins, right? So we can either, first of all, make some depletion of our serum, just have our proteins on a column, pass our antibodies through this column, and only ones which are not bind to those antibodies, those proteins will be possible to use. Or we can use so-called monoclonal antibodies. Monoclonal antibodies are usually mouse antibodies, and they are made this way. We have some lymphocytes from spleen, and of course, one lymphocyte produces only one type of antibodies, right? I mean, one clone of antibodies, of course, not a type. And we can make those cells immortal by fuse them with some contact cells. And we can use terminal cells which have some deficiency, which is not possible for them to grow on some medium. And then we can use chemicals which made lymphocyte which wasn't fused to die, and those cells which do not fuse with our lymphocyte also die. So it's just selective medium. And what we have hybridomas between our lymphocytes and those immortal cells. In fact, they could be used for a lot of passages without any considerable problems. And you can use them for secretion of these antibodies. And since all these cells were provided from only one hybrid of this one lymphocyte and one cell, it will synthesize only one clone of antibodies. And then we can screen which antibodies can bind to which part of our protein. And we can even raise antibodies which can be selective for either some post-translation modification of given protein, so you can recognize not only a protein, but only protein which have some phosphorylation on some certain part of this protein. Or you can even raise antibodies which can recognize proteins of specific folding. So monoclonal antibodies actually can be used a lot. And what we would need for our Western blot, we would need also some way to detect bound antibodies, because antibodies are colorless and we need somehow make them visible. It is possible to conjugation with, for instance, some enzyme like horseradish peroxidase. It can be used by conjugation with some fluorochrome, and we can actually visualize it with special machines. And most of the time we will use such a construction of primary and secondary antibodies, where your primary antibody is able to recognize some of your proteins or even some post-translation modification of your protein. And the secondary antibody can recognize the first antibody. For instance, if you have different animals and you can purify antibodies, the whole pool of antibodies of first animal, let's say mice, and you can use those antibodies as an immunogen for different species. For instance, a rabbit. Because antibody is a protein, it will cause immunogenic response, and you will have a lot of polyclonal antibodies which will recognize those antibodies from mice. And you can use them in this way. So there is a target protein, there is a primary antibody, and a secondary antibody which is conjugated with some label. Why this is usable? What do you think? Why not to use labeled primary antibodies? There are at least two reasons.
Because the secondary antibody, the creation of secondary antibody is easier to sequence of primary antibody than to directly to the protein. So all primary antibodies will be linked with the secondary.
Close enough. Primary antibodies can be monoclonal. Secondary antibodies always polyclonal, and they can find different places within some antibody. You get some amplification of your signal, right? And also it's easier because you can use the same primary antibodies for different detection systems. You could use diamidobenzidine visualization, for instance, chromogenic with the use of horseradish peroxidase. You can use secondary antibodies with fluorochromes or with some other labels without any problems, right? You have your precious primary antibody and a library of secondary antibodies you can use for your research. So, what we next to do then? I think it's on the protocol, unfortunately, without any pictures. But still, we have our membrane with our proteins. The problem is, if we'll just pour our antibodies on this membrane, what will happen? Will those antibodies only specifically bind to epitopes, or can there be any other interaction?
Yes, can be other interactions.
Other interactions. Our PVDF membrane actually bind all the proteins, including those antibodies, because of hydrophobic, hydrophilic ways. And we need to somehow make our antibody to bind only specifically, right? So we need to so-called make a blocking step to block our membrane. To put here some proteins which we are not interested in, which will block all the parts of the membrane which can catch non-specifically our antibodies, right? Like we can use casein, we can use actually secondary antibodies without any label. Also possible to use them. Anything that is that have similar characteristics to what we will be using next. And then when we form our antibodies, they have no way to non-specifically bind to our membrane or proteins, and only if there is specific epitope that we could recognize, they will be on this membrane, and we will see only our signal. So I wouldn't be reading this protocol. It's well, easy enough. And if you use just fluorescent labeling, you could put your secondary antibodies after washing and put your membrane into imager to just see the fluorescence. If you are using chromogenic labeling, you will need to put some special chemical agents which can be converted by this enzyme into insoluble form, and this insoluble form should also have some stain, and you will be able to see this stain on your membrane, and those patches will be actually the localization of your protein of interest. So that's all about Western blot. Just small slide about so-called Eastern blot. It's not widely used. It's a way of seeing some post-translation modification of your proteins, and what we already discussed with different dyes, which can recognize, for instance, phosphorylation and all the protein at all, also can be considered as such an Eastern blot. But there are special chemical agents which can chromogenically stain some of such modifications. So Eastern blot is not used much. It's just a kind of Western blot which allows you to see post-translational modification. We actually can use some antibodies to do the same work, right? So it's possible to do so. There is other way to see the concentration of your protein, a so-called ELISA, enzyme-like immunosolid assay, which uses a similar approach. Like if we can cover our plate with proteins of interest, we can use our antibodies even with label. We can even use primary antibodies with label, and then make some random washing. And only if the protein of interest is blotted on our wells, there will be any signal. And as you can see, it's possible to do, it's possible to quantify. There is a really nice curve here with linear range, so we can actually get the concentration. Actually, thanks to COVID, this technology is now known by a lot of persons, and ELISA can be done in many ways. We can immobilize not our protein of interest, but some antibody which can catch our protein of interest from the solution. And this way it can work even differently. We can actually make it this way. For instance, we can have already recorded antibodies on our wells, which recognize some part of our antigen. This way we can get it from the solution. We do not need to coat our wells with this antigen. And then we can recognize it again. Now with some label, for instance. Another way of ELISA. So there is at least 5 different types of ELISA which allow you to either detect protein which is on the well, which is in the solution, with some additional rounds of amplification, and so on and so on. So you can quantify your protein of interest in solution. So, to sum up, we've been able to see how we can how we can separate out complex mixture of proteins using different electrophoresis techniques. Some of them are even in 2D format, so we get even more information. We've been able to stain the gels to see the whole protein, and we were able to find some proteins of interest on those gels as well. Unfortunately, today I'm almost out of time, so some protein chips and some 2D electrophoresis, which is mostly used today, which allows you to see as much proteins on the same gel, will be covered next time. So if you have any questions, please ask them. If not, friends, thanks for your attention.
Thank you.
I do understand that this part of gels is quite boring. But unfortunately, I don't know how to make it less boring. It's just a lot of information without anything that can be animated or somehow described in other ways. So,
It was less boring than usual, I can say.
fine. Because it's the most boring part of this course, and actually, I think the most boring lecture of all lectures that I. So, well, thank God we lost it.